Hello everyone, I am Nivedita Jana from Ambika Prasad Research Foundation. Welcome to all of you to our channel, Biodiversity and Conservation. So today, in this part of environmental science, we are going to discuss about the nitrogen cycle, its method and mechanism. So let's start. So nitrogen cycle, it is an example of gaseous biogeochemical cycle. The main reservoir of nitrogen is the atmosphere and is also the most abundant element of the atmosphere. The nitrogen cycle represents one of the most important nutrient cycles found in ecosystems, and nitrogen, an essential element of all protoplasm, is a required nutrient for all living organisms to produce a number of complex organic molecules like amino acids, the building blocks of proteins, and nucleic acids, including DNA and RNA. So the main steps in the biogeochemical cycling of nitrogen are presented in this figure. The nitrogen gas, the nitrogen fixation, then the organic nitrogen production, and then ammonification to ammonium, and then nitrification and nitrite formation, and then denitrification, and again formation of nitrogen gas. So it is showing the basic steps in nitrogen cycle, and the nitrogen fixation and denitrification are non-reversible processes, while nitrification is reversible. Then nitrogen fixation, uh, this flowing diagram showing various methods of nitrogen fixation. So nitrogen cannot be utilized by the plants in its native elemental form. It must first be combined with other elements such as hydrogen, carbon, or oxygen before it can be used by the plants. The process is known as nitrogen fixation, chemically referred to as nitrification, and is achieved primarily by some biological organisms and also by non-biological methods. So the nitrogen fixation is performed by two methods. One is physical or non-biological, and another is biological. In physical and non-biological nitrogen fixation, the industrial and natural are two elements. And in biological nitrogen fixation, there are two types of process. One is non-symbiotic and symbiotic. And again, the symbiotic is divided into two types. One is non-legumes and another one is legumes. So the biological nitrogen fixation, it goes on in both the autotrophic and heterotrophic strata of the ecosystem and in both aerobic and anaerobic zones of soils and aquatic sediments. And it is achieved by symbiotic and non-symbiotic means. The symbiotic nitrogen fixation occurs in plants that harbor nitrogen-fixing bacteria within their tissues. Each of these is able to survive independently, but life together is clearly beneficial to both, and only when they come together in a symbiotic relationship can nitrogen fixation takes place. So the most familiar examples of nitrogen-fixing symbiosis are the root nodules of legumes. In these leguminous associations, the bacteria usually are rhizobium species, but the root nodules of soybeans, chickpea, and some other legumes are formed by small-celled rhizobia termed radiorhizobium. Nodules on some tropical leguminous plants are formed by yet other genera. Rhizobia are gram-negative bacilli that live freely in the soil. However, they cannot fix atmospheric nitrogen until they have invented the roots of the appropriate liquid. And the non-symbiotic fixation of nitrogen is done by free-living nitrogen fixers, which includes soil and water bacteria, blue-green algae, and yeast. So, the nitrogen-fixing aerobic bacteria, for example, Azotobacterium and anaerobic bacteria, Clostridium, are distributed widely in soil, fresh water, and marine waters. Phototropic purple non-sulfur bacteria like Rhodopseudomonas and phototropic purple and green sulfur bacteria such as Chromatium, Thaocapsa, etc. are also involved in this process. These bacteria fix nitrogen of the soil air in their own bodies in the form of amino acids from which they build their own proteins. When these bacteria die, their proteins are released into the soil. And in aquatic ecosystem like freshwater and marine, the nitrogen of atmosphere is fixed by numerous blue-green algae such as Anabena, Otoshina, etc. In India, rice is grown year after year on the same land without adding fertilizers. During monsoons, luxuriant growth of blue-green algae occurs as gelatinous masses in the waterlogged fields. They fix up atmospheric nitrogen in the soil. Alocina fertilissima, the dominant alga of rice fields, is the most powerful nitrogen fixer. And the certain yeast cells and mycorrhiza fungi fix up a very small quantity of atmospheric nitrogen. So let's discuss about the symbiotic nitrogen fixation versus non-symbiotic nitrogen fixation. So in symbiotic nitrogen fixation, a part of a mutualistic relationship in which plants provide a unique and fixed carbon to bacteria in exchange for fixed nitrogen, where a process of biological nitrogen fixation performed by a group of autotrophic bacteria living free in the soil, either aerobically or anaerobically and not dependent on plants in non-symbiotic nitrogen fixation. And symbiotic nitrogen fixation bacteria live in a mutualistic relationship with plants, where non-symbiotic nitrogen fixation bacteria are free living in the soil. In symbiotic nitrogen fixation, ammonia, amino acids, and ureates are formed, where ammonia, nitrates, and nitrates are formed in non-symbiotic nitrogen fixation. Symbiotic bacteria produce nitrogen for its host, and non-symbiotic bacteria make nitrogen available in the soil. Symbiotic nitrogen fixing bacteria, for example, Rhizobium trifolium, Rhizobium leguminosum, and example of non-symbiotic bacteria is Agiobacter species and Granulobacter, etc. Then coming to the mechanism of biological nitrogen fixation. So biological nitrogen fixation can be represented by this equation in which 2 moles of ammonia are produced from 1 mole of nitrogen gas at the expense of 16 moles of ATP and a supply of electrons and protons. This reaction is performed exclusively by prokaryotes like the bacteria and related organisms using an enzyme complex termed nitrogenase which catalyzes the splitting of N2 This enzyme consists of two proteins, an iron protein and a molybdenum iron protein. The reactions occur while N2 is bound to the nitrogenous enzyme complex. The Fe protein is first reduced by electrons donated by the ferredoxin. Then the reduced ferrum protein binds ATP and reduces the molybdenum iron protein, which donates electrons to N2, producing Hn is equal to NH. In two further cycles of this process, Hn on NH is reduced to S2N single bond NH2, and this in turn is reduced to 2NH3. Depending on the type of microorganism, the reduced ferredoxin which supplies electrons for this process is generated by photosynthesis, respiration, or fermentation. Nitrogen fixation is energy-expensive process, and much energy is required to break the triple bond of molecular N2 so that it can be converted to two molecules of ammonia. For biofixation by legume motile bacteria, some 10 gram of glucose from plant photosynthesis is required to fix 1 gram of nitrogen. Free-living nitrogen fixers are less efficient and may require up to 100 gram of glucose to fix 1 gram of nitrogen. Similarly, a lot of fossil fuel energy has to be expended in industrial fixation, which is why nitrogen fertilizer is more expensive than most other fertilizers.